synapses, action potentials, nerve potentials, super cool stuff today. You've probably seen a picture that looks something like this at some point in your life where there's like impulses being sent through nerves and your brain cells are talking to one another and it's telling your body to do things. If we actually look at a nerve cell, it looks like this. So the anatomy of a nerve cell, you have this cell body here that contains the nucleus. It's still a cell, it still needs DNA. So it has this nucleus here, and if you look, this body extends and we have these little things called dendrites. Dendrites receive signals, and then when dendrites receive signals, notice here how it says the direction of the nerve impulse is down this way. So I receive some signals and they pass along through the axon. This part of the nerve cell is called the axon, and that's the direction of the nerve impulse. Then when I get to these things here, these are axons. Still, this whole thing's the axon, but it kind of breaks up into these multiple little pieces here so that the signal can be sent out through these little arms. So receiving the signal, traveling down, and then the signal is released. And really what's happening in this picture is that multiple of these are connected together. So one of these dendrites from one of these neurons is accepting a signal. Then I'm getting that signal passed through that, and then it gets released to another one, and then that's the firing. So we're going to zoom in on this red circle here. That's called the synapse. So this is when I have the axon of one releasing a signal being accepted by the dendrite of another. So this is how these nerve cells communicate with one another. This is called the synapse, this space in between, and the type of communication we're seeing here is paracrine because these cells are close but not quite touching. So again, they're multiple neurons connected to one another, but not actually touching. And then the axon of one sends a signal to the dendrites of another, and this is the reception happening here, where that ligand, that chemical signal, is binding to a receptor. We're going to see what happens after that binding occurs. But in order to do that, you have to know a little bit more about what these neurons look like on the inside. So you can see here I have some ions, I have some charges. The inside of a neuron has mostly potassium ions, this K+. There's some other stuff happening there, but this is the most important thing, these K+. The outside, however, has Na+. So even though Na and K are both positively charged, notice how many more sodium ions are on the outside of the cell. Because the outside of the cell has a higher positive charge than the inside, then we say that the outside of the cell is positively charged and the inside is negatively charged, because there are more positive sodium ions outside than there are positive potassium ions inside. This is what's called a resting potential. This is normal when no nerves are being fired. And this happens at -70 millivolts. So -70 millivolts is the term for the resting potential. And this is just what it is normally, because I have that negative charge inside due to this uneven distribution of charges inside and outside of the cell. So I have more positive charges outside, so the outside is positive, inside is negative, specifically -70. This is what's happening normally, before any nerve impulses are being sent. In order for a nerve impulse to be sent, something special has to happen. First, as always, reception. So this is that other neuron, and these are what are called, neurotransmitters. I don't know why I blanked on that. So these are called neurotransmitters. An example would be like glutamine. You just need to know neurotransmitters as a general term. So remember that these receptors and this ligand, or this chemical signal, specifically a neurotransmitter and action potentials, is a nice shape to fit nicely in this receptor. So when that binding occurs, that's reception. So this allows for sodium to flow through that into the cell. Okay, so now I'm getting more positively charged inside the cell. So my potential here is going to start to slowly increase. If more neurotransmitters bind, so I'm getting even more binding, that means that I'm going to be getting even more sodium flowing into the cell. And again, that means that the inside of my cell is getting less negative and more positive. So I'm increasing that potential. Now this negative 55 is something that's very, very important. The negative 55 is what's called the threshold, and that'll make sense in a second. Once we reach the threshold, then this protein here kind of pops its lid. It is a voltage-gated ion channel. So break that word down, voltage-gated. A gate requires a key. And in this situation, the key is some sort of voltage. So in order for this to open, it needs to have reached that threshold. That's why we call it a threshold. So at negative 55, this guy pops its lid. And when that guy pops its lid, it's specific to these sodium ions. So now even more sodium ions are flowing in, which means that I'm going to continue to increase this overall potential within the cell. And the important thing to note is that this is propagating. It's like a domino effect. So they all don't open at the same time. Because sodium ions are flowing in here, I mostly have that increase in charge here. Now, when collisions are happening and those positive charges are moving down this way, now boom, this one opened up via the key. But it's not yet negative 55 here. So this one's nice and closed. But as I continue moving sodium in, now this one's at negative 55. So it pops its lid. This one pops its lid. And now I'm getting more Na into the cell. And what's happening here is instead of having a negative charge on the inside, I'm eventually increasing that charge. And these are now becoming positively charged on the inside and negatively charged on the outside. But it's like a domino effect. So they don't all pop and move in at the same time. It's a domino sort of effect. So I am reaching that threshold, pop, 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 moving more in, but I'm reaching the threshold as I move down that neuron. Okay, so now I have reached another very important kind of key here. So these are my voltage-gated sodium channels. These green ones here are my voltage-gated potassium channels, meaning they transport potassium and they're voltage gated. So they also require a key in the form of a voltage, and specifically for these channels, they need this voltage here of positive 30. So once I reach positive 30, these ones are going to start popping their lids. And they pop their lids this way, and potassium moves out because of the concentration gradient that we learned about last try. So see where all of the potassium is. The potassium is going to want to move down its gradient, so the potassium is going to move out. And now the opposite kind of thing is happening, and instead of the inside of the cell getting more of a positive charge, the inside of the cell is losing its positive charge, so it is becoming more negative again. So this is happening down, down, down in like a domino kind of effect. And an interesting thing is that so much potassium leaves that I actually end up lower than I started. I end up down here at a negative 90. And this is our body's kind of safe. It helps us ensure that we're not firing too rapidly, so that we have to kind of wait a minute before we can fire another nerve impulse. This is something that doesn't happen in individuals that have epilepsy, and that's why they are so much more easily triggered by signals. They're sensitive to light, they're sensitive to all of these things because their body does not reach this negative 90. It doesn't allow them to do a reset before they can just fire, 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 fire. For us, if you don't have epilepsy, our body pushes out enough of that potassium through those voltage-gated ion channels that I have become more negative than I started out with. So that might not sound very good, but this is where the sodium-potassium pump comes into play. If you remember the sodium-potassium pump, it is a form of transport protein that requires ATP that pumps sodium and potassium into and out of the cell. Specifically, the sodium-potassium pump transports 3 sodium out and 2 potassium in. And this is great for our cells because remember, we started out with more potassium inside. And then due to these ion channels opening, I reversed that. And now I have all of the sodium in and all of that potassium out. So once I get to this threshold here, this new kind of voltage that allows me to do a reset, the reset is accomplished from the sodium-potassium pump. So the sodium-potassium pump pumps out 3 sodiums and pumps in 2 potassium. And okay, so I still have more positive charge outside, and that's why it doesn't increase it that much, but it brings me back up here to this 70, which is my resting. And then that's the reset. Now more neurotransmitters can recept here and cause a signal, and then I need to reach the threshold again, and then boopity boopity boop. It is a domino effect. Super, super cool. We're going to look at the parts of this graph really quickly. So normal resting potential, that is called polarized. Our cells are polarized, our nerve cells, because they have an uneven distribution of charge, because I have more of that sodium outside the cell and more of that, or less of that potassium inside the cell. Remember, it's reversed from this when I first start. That's polarized. When those sodium ions start flowing in due to that neurotransmitter bonding and sodium flowing in, and then the threshold being reached because of it reaching negative 55 and now even more sodium flowing in, this step here is called depolarized. And that's because I am starting to even out those charges. 0 crosses somewhere in between here. So I'm polarized and then I'm depolarized, and then once I reach this, I am repolarizing. That's kind of nice that I'm polarized, depolarized, so I'm doing the opposite, and now I'm coming back, so I'm repolarizing. This little part here where I go lower than I usually go, that's hyperpolarized. So I'm even going beyond what I started out with. So hyperpolarization is something that individuals with epilepsy can't do. They can't do the reset, so they just fire, fire, fire. We do this reset where we're hyperpolarized, and then the sodium-potassium pump helps us get back to normal. So to summarize those steps. So first we have this resting potential, where we are at negative 70 millivolts because I have a lot of sodium ions outside the cell and some potassium ions inside the cell. The neurotransmitter binds, allowing that sodium to flow inside the cell. As more sodium flows inside the cell, that threshold potential of negative 55 millivolts is reached, and that opens up the voltage-gated sodium channels, and even more flows in. That's depolarization. So I was polarized at negative 70, those sodium started flowing in, increasing the charge up to negative 30, or positive 30. When that positive 30 is reached, those voltage-gated potassium channels open, and potassium starts to leave the cell. And because that positive charge is leaving the cell, it's losing that positive charge and it's becoming repolarized. So much of that K plus left the cell that the potential ends up being lower than what it started at. Negative 90 versus the negative 70. This is hyperpolarization and allows us to rest our nerve cells before firing them again. The sodium-potassium pump is what does that resetting, and it transports 3 sodium back out, which is what I initially started with, and 2 potassium back in, which is where they normally started, until the cell reaches that negative 70 millivolt resting potential. And then it's ready for another impulse. Done!